Because, whoa, mutation in my DNA. Wait 1 second. Okay, now, DNA. Okay, I know it's DNA because I've got deoxyribose sugars, sugar phosphate backbone. I got my bases sticking out, and look, guanine to cytosine adenine to thiamine. So thiamine is connected to a deoxyribose sugar. So it's definitely DNA. Double-stranded molecule. This is my messenger RNA. I went ahead and made it ahead of time faster, okay? But look, I can tell it's RNA, ribose sugars. I got uracil instead of thiamine. Watch. This model is designed so that you can't put uracil on a deoxyribose sugar.
Oh.
Isn't that cool? And you can't put thiamine on a ribose sugar because they don't go on that. But when I put them on there, look at their design. Do they act the same? Yeah. So uracil acts like thiamine, so it doesn't really matter that much. It's just you need to understand that it's uracil on RNA instead of thiamine. Okay? Oh, I got a mutation. Okay. So now watch. I take the DNA. I want this gene. I want GTCC, right? I want that sequence. So I need to make a messenger RNA. So I separate the DNA and I make a messenger RNA transcript. When I make that messenger RNA transcript, I make it off of the opposite strand of the gene that I want. This opposite strand is the template or the antisense strand. Very good. Okay. And this is the sense strand. Okay. So this is what I want, this sequence. So I got GTCC, I end up making GUCC. Is that the same?
Yeah.
Yes, because U acts like T. You got it? All right. Now, I make that messenger RNA. Let's say it doesn't need any post-translation, transcriptional modification, so it's all ready to go. And my DNA stays back inside the nucleus. Wait, where am I? Inside the nucleus, right? Okay. And then I get my messenger RNA attached, and the ribosome is the silver tray. Now, I read and I say, okay, the ribosome's reading GUC. I need an anticodon that matches, and this transfer RNA comes in from the cytoplasm.
Can I ask a quick question? So
Yeah.
now this is the translation part?
Yes.
Okay, fine.
So I transcribed, now I'm translating. So now I've got my GUC. I need an anticodon. This is my transfer RNA, and it's carrying the yellow amino acid. It turns out that GUC codes for the yellow amino acid. You with me? All right, so I got it lined up exactly on the P side here. I got the A side open. You with me? So the A side, you come in, and as you're trying to get lined up, you get these two amino acids so close to each other, and they just happen to be right on top of peptidyl transferase, the enzyme, on the 60S subunit needed to make that peptide bond. You lose the hydrogen from the amino group. You lose the OH, the hydroxyl, from the carboxyl group. You with me? And you end up making water. Okay? And boom, peptide bond. Okay? Now, I've shifted the ribosome. Now the P site's right here. So do I need this transfer RNA anymore? It's done its job. It delivered its goods. So it leaves, goes out in the cytoplasm, picks up another yellow one. Never a green one, never a red one, never a blue one, always yellow. Because that way you make sure you match up to the correct codon with the correct amino acid. Now the A site's open, so I do it again, and it's getting boring now because it's the same process. Peptidyl transferase pulls off the hydrogen and the hydroxyl, makes that peptide bond, dehydration, condensation, shift everything over. Now the P site's right here. Okay, A site's over here. So do I need this transfer RNA? Nope, delivered its goods, goes out in the cytoplasm, and picks up another green one, only a green one. That way you can keep things right. Then you go you do last codon, let's say it's a UAA, stop codon, right? Okay. You get to the UAA, all right? That means you come in with releasing factor. Releasing factor binds, okay? Yehuda gave me an idea of doing that. So it's releasing factor binds, okay? And then what happens is now it causes everything to pull apart. So you get the last of the transfer RNAs leaving, going out and get a red one. Only a red one. And your messenger RNA, if you only need to make one of these, what are you going to do with this messenger RNA? Destroy it. Otherwise, you start all over again. And if you look right here, I just made what?
Polypeptide bond.
Polypeptide. And the polypeptide has yellow, then green, then red amino acids. If it was yellow, then red, then green?
Different.
Different. Yeah. So it's really important you get the right sequence. And it's